previous lecture, we have discussed about the general overview and the basic principle of centrifugation technique. If you could recall, we have discussed about the various factors which are involved in the centrifugation of a particular particle. Now, these depends on like the size or we can say mass of the particle, density of the particle, whether it deviates from a spherical molecule, that is whether it is ideally a spherical molecule or aspherical molecule or non-hydrated or hydrated molecule. All these factors play an important role in the sedimentation of or the sedimentation rate of a particular particle in this under the influence of centrifugal field. Also we have discussed about the sedimentation coefficient, where it is expressed in terms of Swedberg units, and we have discussed about how different types of particles say biomolecules or subcellular organelles has different sedimentation coefficient value in terms of Swedberg unit. In this lecture, we are going to extend our discussion to the types of the different types of centrifuges, types of rotors which are used in these centrifuges, then care and safety aspects of centrifuges as well as rotors used in them. So, we will be discussing all these things in coming lectures. In this lecture, we are going to discuss about different types of centrifuges used for various applications. Now, centrifuges are the devices used in a variety of scientific and technical applications. Now, these centrifuges, what they do is they in particular spin carrier vessels, or we can call them centrifuge tubes actually, at a very high rotational speed, or you can say at very high applying very high centrifugal force. Now, the centrifugal force generated in here is proportional to the rotation rate of the rotor in terms of RPM, and also the distance between the rotor and the centrifuge tube. So, both these factors affect, that is how fast it is rotating, a tube is being rotated in terms of revolutions per minute, and what is the distance between the rotor center, that is the axis of rotation, and the centrifuge tube. Now, there could be like each centrifuge which could be of different types, as we are going to discuss about them. They can carry multiple types of rotors of different sizes, and so that the volume of sample which could be analyzed or which could be processed is like you have flexibility in choosing the centrifugation conditions for a particular type of analysis or a procedure. Now, the various applications that can be performed depends on the relative centrifugal field. So, you can see here there are some examples of a very small microcentrifuge or microfuge, you can say a quick spin type centrifuge. There is a tabletop centrifuge which is refrigerated centrifuge, and these are little higher floor model centrifuges. So, there could be various kinds of centrifuges for various kinds of applications with lot of flexibility. Now, in terms of the kind of speed, that is in revolutions per minute, and the maximum relative centrifugal field which could be carried, the centrifuges can be classified into say low speed centrifuge, high speed and ultra centrifuges. Now, low speed, like as you go along with the various parameters given here, if you have a low speed centrifuges will have revolutions per minute around 10,000, high speed will have 28,000 revolutions per minute. These are approximate values, and ultra centrifuge will have say 100,000 revolutions per minute. So, what you see approximately, and so corresponding RCF values are also given in here which is like 7,000 G force 100,000 or 800,000 G force approximately. So, what kind of applications could be, like pelleting applications could be performed with these kinds of speeds here. If you see there are, like these are listed in here, say bacteria can be pelleted at all these, but you do not really require ultra centrifuge for this, though you can do it, but it could be done at low and high speed centrifuges. Likewise, animal and plant cells can also be pelleted at up to high speed centrifuges. Nuclei, then certain precipitates. Now, in nuclei you can always use any of these. Certain precipitates like high speed centrifuges are quite good for that, maybe low speed is not enough, and you can also do it like mostly they are not used, like in brackets we have put in it can be done, but not usually used for this purpose. Now, if you go further down here, you can see that the membrane fractions, ribosomes, certain macromolecules, viruses, these all require ultracentrifugation. So, you can see that as the size goes smaller, you require a much higher relative centrifugal field for sedimentation of these molecules, particularly biomolecules. You require very high speed, or in terms of RPM, or in terms of relative centrifugal field. So, this just summarizes the kind of applications which can be done on low, high or ultra centrifuges. All right. So, before we go on each type of centrifuge, the working of a centrifuge essentially requires a material to be spun in centrifuge. So, whichever material needs to be spun is placed in a centrifuge tube, which is then placed in a rotor. Now, rotor and this I have shown you earlier in earlier lectures also. Now, rotor is generally a dense metal which can dissipate heat and could take the stress of because of centrifugal force. It has sufficient mass that it can generate momentum, and it can spin without any problem. Now, work they could be like centrifuges where you require vacuum, particularly for ultracentrifuges, and many of the centrifuges are refrigerated also, not all of them, but nowadays many of these centrifuges are have refrigeration system, and this reduces the heating caused by frictional forces as the rotor spins. Now, rotors are usually stored before you are going to start an experiment. You can store them in a refrigerator or refrigeration unit, so that they could be near the operating temperature. Uh, Also, many macromolecules they could be damaged, or they could may not remain in their active form. So, many times refrigeration is required when you are working with biological molecules. Now, as centrifuges come in all shapes and sizes, and the rotors also vary, the universal and transferable unit of centrifugation here will be centrifugal force, like earlier also like we were discussing that ultimately it is a relative centrifugal field which needs to be taken into account, and then you can through that like through a nomogram we have shown you, you can calculate the revolutions per minute, and then whatever centrifugal force is required, that needs to be should be applied on the particular material for a particular application. Now, there are many different types of centrifuges are available, and they will include variable features actually. Some of might have like say there is a maximum speed of sedimentation which a particular centrifuge could achieve. There could be presence or absence of vacuum, like I said, ultracentrifuges in particular have to have the rotor chamber has to be evacuated. Temperature control or refrigeration is required in many of them, as we have discussed. Then another important factor is how much volume of the sample could be taken by particular rotor type. That means, what is the capacity sample capacity or the volume of sample and a particular capacity of centrifugation tubes. Like for example, if you can put 5 tubes, what is the volume which could be kept in there, and we will be discussing these aspects as we go along. So, let us start with different types of centrifuges here. Now, small bench centrifuges, these are very simple, these are very simple and least expensive centrifuges, and they have many types of designs actually nowadays available. Now, these are like when you say small, you can simply put them on your bench top, they are small, they could be put anywhere, and they are for smaller volumes actually. So, they are used to collect small amounts of material and that sediments rapidly. For example, it could be cells like yeast cells or RBCs that could be pelleted or collected. Maximum speed is 4000 to 6000 revolutions per minute, which could be achieved in here, and corresponding relative centrifugal field is around 3000 to 7000 G force. These can operate at ambient temperature, but certain centrifuges like latest ones might have refrigeration systems here to keep rotors cool and prevent denaturation of biomolecules, particularly proteins. Now, these small centrifuges here, they are also available with more speed or maximum speed, as we have seen 4000 to 6000 revolutions per minute for certain centrifuges, but they are available as in a speed range of 8000 to 13,000 or 14,000 revolutions per minute also, and maybe more also, and the relative centrifugal field may go up to 10,000 G. So, lot of different kinds of small bench centrifuges are available for rapid sedimentation of small amount of materials. Now, these are very useful for sedimenting very small volumes, like I said, up to say 1.5 ml. So, they are sometimes called microfuses, or there is a rotor which contains centrifuge tubes of around 1.5 or so, and so it is for mostly small volume. So, these are examples here. If you can see this is a very small centrifuge, that is small bench type centrifuges, and this could be containing only rotor, which can be small tubes are in there, or there could be centrifuge tubes, rotors are also available in here. So, small bench type rotors are routinely available in lot of different research and teaching labs. Then there are large capacity refrigerated centrifuge. As it says, large capacity means it can take larger amount of sample or more volume of sample. Maximum speed of these centrifuges could go up to 6000 revolutions per minute, and maximum RCF corresponding to that or relative centrifugal field is 6500 G. These are refrigerated, so rotor chamber is refrigerated and large capacity. So, they can carrying capacity is much higher than small bench type centrifuges. There are interchangeable rotors available for this, like there are swinging bucket rotors and fixed angle rotors for these centrifuges. We will be discussing swinging bucket and fixed angle rotors in a later chapter, and separation could be achieved in 10, 50 or 100 ml tubes. Now, these are different, like it can go up to total volume or total capacity could be say 4 to 6 liters are available in these, but there are different capacities available. Now, when you are working on this and we are spinning these tubes here, we said, accurate balance needs to be done, so that these tubes are balanced and rotor imbalance is not there. And they used to they pellet a substance and they sediment rapidly, like say for example, RBCs or certain bulky precipitates or say yeast cells, nucleic nuclei and chloroplast, a lot of other things could be worked upon here for certain applications. This is a typical high capacity refrigerated centrifuge, as you can see, and this can carry like 4 to 6 liters of material, and these could vary in capacity, and various types are available in this range. All right, so we move on to high speed refrigerated centrifuge. Now, high speed refrigerated means they can go to a very high speed, and maximum speed could go approximately up to 25,000 revolutions per minute, and the relative centrifugal field corresponding to that is around 60,000 G. So, now you can see that why one must work with the relative centrifugal field rather than RPM, because when you have lower RPMs, they are almost equal to the RCF value, but here as it goes up, you can see 25,000 revolutions per minute, and RCF is quite a lot here. 60,000 G force is applied on, in high speed refrigerated centrifuges. The capacity here is not like high capacity refrigerated centrifuge, but it could go up to 1.5 liters or so, and this also can take both fixed angle and swinging bucket rotors. They are interchangeable, like you can put either of them on the drive shaft and screw them, and then both could be worked upon. Of course, the speeds of swinging bucket and fixed angle rotors will be different, and that could only be known by specifications of the rotor. Now, these are used to collect say microorganisms, cellular debris, larger cellular organelles, protein precipitated by ammonium sulphate or so. So, a lot of applications are there, even differentiation separation of say nucleus, mitochondria or protein precipitates, large intact organelles, etcetera could be done. Now, so there are a lot of applications of high speed refrigerated centrifuges, but they cannot be utilized for or they do not have sufficient centrifugal force is not generated here to effectively sediment like viruses. If you could recall in the table, for viruses or ribosomes or smaller organelles, you require ultracentrifugation. So, high speed refrigerated centrifuges are good enough for many applications or sedimentation of many different kinds of materials, but there is not like they are not good for say viruses or ribosomes or other smaller organelles. It is a very typical high speed refrigerated centrifuge. They could be like all these centrifuges could be obtained from different companies, a lot of makes are available, and one can compare and acquire one or many of them to work upon. Now, as we were seeing all these centrifuges, these were simple centrifuges where you keep your volume and work upon them, but there are also other centrifuges like continuous flow centrifuges. Now, continuous flow, as name suggest, there is a continuous flow of material from there is inlet and there is an outlet. These are relatively simple centrifuges and they could go to high speed. So, these are high speed centrifuges. Now, rotor is quite different here. It is long tubular and non interchangeable rotor, and the particles or the material which is being used in here or being processed in here, particles suspend in a medium flow continuously through the rotor. So, as medium enters the rotatory rotor chamber, so particle will sediment on its wall actually, and excess is clarified, medium overflows through an outlet port. So, there is an inlet port and there is outlet port. As the medium containing the cells or other material particles enter the rotor, that material on the basis of centrifugal force will pellet onto the walls or sediment onto the walls, and medium is overflows through the outlet port. So, this is done for very large volumes, and major applications are like harvesting of bacteria each cells from very large volumes of cultures, like say a few liters to many like 100 liters or 500 liters cultures are used. We will be discussing about continuous flow centrifuges, continuous flow rotors actually later on. This could be a typical centrifuge for that. Now, as we have seen in our table, the very high speed centrifuges are ultracentrifuges, which could achieve very high speeds, like say in terms of 70,000 rpm or above actually. So, the ultracentrifuges are made for both preparative and analytical applications actually. So, you can do analytical applications could be performed in here, like you can analyze a particular purified or partially purified sample for its sedimentation behavior or other properties, and it could be also preparative actually. So, where you can get after the centrifugation you can get material for further biochemical analysis. Rotors are run in a high vacuum. They are like the chamber where rotors are kept. It is evacuated, refrigerated, and it is sealed chamber. So, this requires much higher advancement, or like say there is a armored chamber and all those things for because it is spinning at a very high speed. So, much more sophistication is required in terms of vacuum or refrigeration in here. Now, maximum speed here as it is evacuated, it is not limited by air friction, and the rotors do not have to be aerodynamically designed as compared to other centrifuges. So, these can reach very high speeds in here, and like I said, the maximum speed, angular velocities or revolutions per minute could reach up to in excess of 70,000 rpm, and it could achieve the g force of 500,000 or above actually, and it could be used for membrane fractionation, and we were talking about viruses or ribosomes which cannot be done by high speed centrifuges. They could be pelleted in here, or they could be sedimented in ultracentrifuges. Typical ultracentrifuge, it is a very schematic, it is a simple schematic diagram of ultracentrifuge, and as you can see there are a lot of different things which are important in here. There is a arrangement for vacuum. You can do switches, or these are like controls for vacuum temperature, indicator lights, rotor speed here. These are lid operating. Then there is automatic rotor speed selection unit is here. There is a protective steel shielding. There is a sliding lid which you can put in things in here. There is a refrigeration unit. So, you can say it is a typical ultracentrifuge might look like this actually, and it has lot of different things like I said, protective steel shielding, good refrigeration system, high vacuum conditions, automatic rotor speed selector unit, imbalance unit like imbalance recognition. All these things are available in here. This is like one typical ultracentrifuge here with all these controls and other things. This is a simple diagram for that for an ultracentrifuge actually. There is a heavy duty motor and a lot of other things as we have discussed in here. Alright, now ultracentrifuges can be of two types like we were discussing. One is preparative ultracentrifuge and another is analytical ultracentrifuge. So, let us start with preparative ultracentrifuge. In preparative ultracentrifuge, maximum rotor speed could go up to somewhere around, or maybe more also, 80,000 revolutions per minute, and it can go even much higher in a bench top ultracentrifuges like 150,000 revolutions per minute, generating RCF value of say 600,000 G. So, this is for floor models actually. These are, like I said, these are sealed, evacuated, refrigerated rotor chamber is available in there, and like excessive rotor temperature which might be generated by frictional resistance between air and spinning rotor is reduced quite a lot in here because of evacuation. Uh, there is a temperature monitoring system which is highly sophisticated, which employs infrared temperature sensors and other advanced technology to monitor rotor temperature and control the refrigeration system, because the speeds are so high that it has to have these kinds of advanced modern systems. There is a over speed control system. So, whatever you set that will be maintained actually. There are electronic circuits to detect rotor imbalance and control refrigeration system. So, there are a whole lot of mechanisms so that these centrifuges could be operated without any hitch. There is a flexible drive shaft system in these centrifuges to maximize to minimize the vibration arising due to rotor imbalance. There is accurate balance has to be done within 0.1 gram, and there are sensors to detect imbalance. So, rotor will not run if there is an imbalance. Rotor chamber of high speed and ultracentrifuges are enclosed in a very heavy armor plating, like we have seen protective shielding actually. So, rotor chamber or rotors are kept in a protective shielding, even if something happens it does not escape the centrifuge. There are other centrifuges called air fuse. Actually, these are an air driven tabletop preparative ultracentrifuges which are available, and they are called air fuse also. And these are capable of accelerating a magnetically suspended rotor and accommodating, say, certain like 1.5 or 1.75 mm cube tubes, 175 mm cube tubes, like it could be 7 it could be like 6 or so tubes of around you can say small centrifuge tubes could be put in there, and it is a virtually a friction free cushion of air, and these are in non vacuated chambers actually. So, those could also be utilized, and maximum speed could go to 100,000 revolutions per minute, around 160,000 g force in very short period of time. Like I said, now there are tabletop ultracentrifuges are available, which could go up to 150,000 RPM, and a lot of small samples, volume samples could be for preparative purpose could be processed in those centrifuges. Essentially, it is also important to note that all these like ultracentrifuges are quite expensive and needs to be maintained in a particular lab. So, those things also has to be taken into account. Preparative ultracentrifuges are very useful, but like for we were saying tabletop or small ultracentrifuges, which are also preparative centrifuges. These are also very important, and small volumes are required, and you can do high centrifugal force could be applied, and a lot of biochemical and clinical studies could be done on there. So, here what is done is you can do separation, isolation and purification of lot of things like whole cells, subcellular organelles, chromatins, nucleic acids and other things. Macromolecules or ligand binding kinetic studies could be performed, like say steroid hormone receptor assays, separation of the major lipoprotein fractions from plasma, and deproteinization of physiological fluids for amino acid analysis, and a lot of other things could be performed on these ultracentrifuges, which are small, which take small volumes. So, this is a typical ultracentrifuge, preparative ultracentrifuge floor model, which is for say A lot of applications can be done on in here. So, preparative ultracentrifugation technique is quite widely used for a lot of different applications. For example, if you would like to separate ribosomes from the cells, then it could only be done in preparative ultracentrifuge. So, likewise many like I told you, viruses or many small subcellular organelles could only be separated in preparative ultracentrifuge. So, there are certain applications where it is a must actually, though like I said, it is quite expensive instrument. All right. So, from preparative ultracentrifuge, let us move on to analytical ultracentrifuge. Now, analytical ultracentrifuges are also very important, and they also operate at very high speeds, which are approaching say 70,000 revolutions per minute, with RCF value of 500,000 G or so. Now, these consist of a motor, rotor inside a particular protective chamber, which is refrigerated and evacuated, and this is like I said earlier, armored chamber, which is you can say protective shield around the rotor actually. Analytical ultracentrifuge, unlike preparative centrifuge, will also contain a system for detection, that is optical system for study of sedimentation characteristics of biological macromolecules and molecular structure and other like to determine say concentration distribution in the sample, or the particularly the sedimentation characteristics are analyzed in here of purified or partially purified samples actually. So, there needs to be a particular type of rotor and particular arrangement, so that optical system could be put in here for real time analysis as the operation is going on. A typical analytical ultracentrifuges, you can say looks like this, and it has all the controls and other things, and also it will contain an optical system here. So, these are just two representation of that. Now, depending on the type of optical systems used in analytical ultracentrifugation, sedimentation in an ultracentrifuge is observed in that particular way. It could be like concentration based or say concentration gradient as a function of radius. In both ways it could be seen, and there are different types of optical systems are available, and analytical ultracentrifuge can come with those kinds of optical system. So, three types of optical systems which are available. One is absorbance optical system. Absorbance optical system displays the boundary in terms of optical density as a function of radius. So, as the like say if you have certain sample, say for example protein or nucleic acid sample, and as you layer it or is it present in the whole of the solution, as it sediments there will be like area where it moves down and concentration increases, and particular peak will be obtained in here. As the concentration will increase, certainly the peak will also will be like different. So, what you do is that measurement of sample concentration will be done, or it will be analyzed at wavelengths which could be from ultraviolet region to visible region, say 200 to 800 nanometer, and particularly nucleic acids and proteins could be analyzed in UV region. Many of these macromolecules may contain strong chromophores, and that could be analyzed, and so they could be analyzed on that basis through UV wavelengths, or like in wavelengths in ultraviolet or visible region, and it could be like the concentration differences could be recorded and they could be analyzed in real time. Then there are alternative Silurian system. Like earlier in absorption system, it was like particular wavelengths are absorbed by these macromolecules, like say nucleic acid absorbs at 260 nanometer or protein absorbs at 280 nanometer, or they might contain certain chromophores which absorb at other certain wavelengths. These peaks or the data could be generated. So, in alternative Solarian system, it detects the changes in refractive index of the solution. So, it will display the boundary in terms of refractive index gradient as a function of radius. So, what does that mean is, when you have taken a solution in a particular sector cell or tube in analytical ultracentrifuge, when the solution is homogeneous, you will not see any boundary, there is no boundary as such, and there will not be any gradient of refractive index. So, nothing, no signal will come. But as the centrifugation starts and the macromolecule or whichever is the material starts sedimenting, then a boundary or a zone is created which moves in the direction of centrifugal force actually. And this boundary which is created, this is detected in terms of refractive index gradient as it moves down the cell. So, here what is done in alternative Solarian system, that is refractive index of the solution, or you can say measurement of sample concentration based on refractive index changes are performed. This could analyze macromolecules which are lacking intense chromophores, or for example, which cannot be seen in absorption system, like polysaccharides or carbohydrates are mostly analyzed with a detector that is refractive index measuring detectors, rather than UV monitor or absorption based detector. These samples require refractive index detector. Now, there is third system which is Rayleigh interference optical system, and Rayleigh interference optical system is like, or it is an improved version you can say of alternative Solarian system, where what you have is that the sensitivity of Rayleigh interference optical system is much higher as compared to alternative Solarian system. And here as the concentration is higher, many systems utilize Rayleigh interference optical system. So, essentially what you have is you have three systems. One is absorption system, where particular macromolecule or a chromophore can absorb the wavelength either in say UV range or visible range, and that will be recorded and analyzed. The data could be analyzed in there. Alternative Solarian system and Rayleigh interference optical system will be detecting the changes in the refractive index of the solution. As far as rotors are concerned, rotors for analytical ultracentrifugation must be capable of withstanding enormous gravitational stresses, and under these conditions a lot of, like a mass of 1 gram experiences an apparent weight of say 250 kg. So, that is kind of a stress here. So, rotor must allow also. So, not only it could take stress, as well as it has to allow the passage of light through the spinning sample. That is, you have to install optical system in here, and some mechanism must be available for this, and also you have to control the temperature. Rotor is solid in this analytical ultracentrifugation with holes to hold the cells that contains samples, and it is suspended on a wire coming from drive shaft of a high speed motor. Tip of the rotor contains a thermistor for measuring temperature. I will show you this in a little while. And the simplest rotor have two cells. One is called analytical cell and another is called counterpoise cell. Now, counterpoise cell have two holes to facilitate the calibration of distances in the analytical cell, and variety of analytical cells are available with volume capacities which could go from 0.02 to 1 ml. There are sector shaped cells essentially in velocity experiments, and these sector shaped cells could be single sector like 2 degree or 4 degree sector shape could be there, or to prevent convection. An optical path length which can vary from say 1.5 mm to 30 mm, usually with 12 mm optical path lengths centerpieces are available. There are so single sector cells could be there. There are double sector with say 2 2.5 degree sectors and optical path length which can vary from 12 millimeter to 30 millimeters. So, there could be various types of cells available. Double sector cells permit us to account for absorbing components in the solvent, to correct for the redistribution of solvent components and measurement of differences and sedimentation coefficient and diffusion coefficients. Counterpoise cell is so designed that when correctly aligned in the rotor, the walls will be parallel to the lines of centrifugal force, like a swinging bucket rotor which is extended outwards. So, there is no accumulation of material against the wall of the analytical cell, and like the sector, like it kind of spreads out as it goes away from the center of the rotation. Analytical cells have upper and lower plane windows of quartz or synthetic sapphire, and these are to withstand very high G force here. Uh, so, rotor chambers have upper condensing lens and a lower collimating lens in optical system. Upper condensing lens together with camera lens focuses light onto a photographic plate or photomultiplier. Lower collimating lens collimates light so that the sample cell is illuminated by a parallel light. So, this is how the arrangement or optical system is installed in here. In more advanced instruments, like I said, photographic detector could be replaced by electronic scanning system, and data could be collected right away and could be analyzed also and seen also at that as the experiment is going on. So, this is a very simple schematic here, if you can go through. If you can see here, this is a slit here, there is a collimatic lens I was talking about. These are this is this is a solid rotor which contains a counterpoise cell and analytical cell, and the this is placed in here with the collimating lens. Light comes from collimating lens, passes through the analytical cell and then collected by condensing lens, that is reflected by mirror, and there is a system of Selenium diaphragm, camera lens, cylindrical lens, and finally photographic plate where peak or the data is being recorded. And like I said, photographic film could be replaced by electronic scanning system actually. You can see this rotor is kind of suspended through a wire here, which kind of balances this, and there is a thermistor here to sense the temperature. There is a cooling and vacuum systems to evacuate the chamber where rotor is placed, and there is armored shielding here, which is you can say armored chamber for rotor. So, what you have is a system where one is rotor is placed in a evacuated, refrigerated and armored chamber. There is a temperature sensor, and this rotor is a solid rotor with two places or two holes where a counterpoise cell and the analytical cell could be placed in. So, and there is a at the below the rotor at another tip there is a wire or there is a thermistor which senses the temperature. There is a well placed optical system which passes through analytical cell and analyzes the sample or the sedimentation characteristics or different applications as the experiment is going on. There is another representation of this whole thing. where you can see that there is a lamp here, aperture, the light is there is a toroidal diffraction grating, incident light detector is here and reflector. There is a sample cell, these are two cells in here of in this particular rotor chamber. There is place for two cells, and here the there is imaging system, and finally photomultiplier tube is utilized in place of photographic film. If you see here, there is a reference cell, and I was like sector shaped, like they are here. If you can see, the diameter or this particular part increases as it goes from one side to another. So, that there are no wall effects or sedimentation of walls actually, and they are like act as swinging bucket rotors only. So, this is how these are placed, and the analysis could be performed in here. So, if we see principles of Silurian optical system for here. So, if light passes through solution of uniform concentration, it does not deviate, like I was saying, if it is a homogeneous solution, there is no boundaries and no deviations, but on passing through a solution of different densities, it is refracted at the boundary between these zones actually, and the optical system records the changes in the refractive index of the solution, which will vary as the concentration changes. So, two Silurian systems are available, one is optical Silurian system called OSS or bright field scanning optical system. In first one optical Silurian system, the deviated light is passed through an inclined Silurian diaphragm and a cylindrical lens, as I have shown you. The sedimenting particle or material in analytical cell forms a boundary between the solvent which is cleared of particles and the remainder of the solution containing the sedimentation material. So, what happens, this boundary behaves like a refractive lens and which results in the production of a peak in the final image on the photographic plate or detector system. Now, peak is an exact record of the refractive index gradient, and area beneath it is proportional to the concentration of the solute. Rate of shift of the peak will give a measure of the rate of particle sedimentation, and after a period of sedimentation, the peak height diminishes and the width increases. And why this is? Because you have a sector shaped cell, as I have shown you that width of the cell is wider at the centrifugal end. So, OSS or optical Silurian system plots the refractive index gradient against distance along the analytical cell, which makes it useful for locating boundaries in sedimentation velocities measurements. Now, in bright field scanning optical system, the light is deviated, or you can say deviated light is interrupted by a knife edge, and resultant image is scanned with a photomultiplier. The resultant refractive index gradient appears as a dark band against bright background. So, this is how through refractive index detector, and it will be same for say absorption system, the analysis is performed. Let me show you on your screen what is going on in here. So, so you have a, like I said, you have a cell or which is somewhere in this shape. So, you can see this, that it is wider at this place than at this place. So, what happens is when this is filled in with homogeneous solution, then there is no refractive index or there is no boundary as such, and so no refractive index is registered actually, no changes are registered. But as centrifugation occurs and centrifugal force is in this direction, the material or macromolecule or whatever it is, it starts moving towards the centrifugal end because of the centrifugal force. So, a boundary is created, and this area which is in here is devoid, now is devoid of the sample. So, this boundary kind of acts as a lens and a refractive index gradient occurs, where you have more concentration in here as compared to this. So, as this peaks moves in here, you will get this peak as it moves. So, the refractive index gradient or it moves also, like peak also moves. So, what you get is, you get, as you start, you get a peak which is sharp, but as you as this peak moves and comes in, this peak will start, the height of the peak start diminishing, and you get subsequently what you get is this peak spreads out, and finally it is quite spread out actually, because of this shape here of the cell. The Cellerian system is the very widely used, likewise absorption system, depending on your application. Now, the third one, Rayleigh interference optical system, like I said, it is like alternative Cellerian system, but it detects changes, you can say it is highly sensitive or more sensitive to detect small concentration as compared to alternative Cellerian system. So, it employs double sector cell, in which one sector contains the solvent and other the solution, and the optical system measures for the differences in refractive index between the reference solvent and the solution by the displacement of interference fringes caused by slits placed behind the two liquid columns. Now, each fringe tracing a curve of the refractive index gradient against distance in the cell. Now, since the position of the fringes is determined by solute concentration, it is possible to measure the concentration of solute at any point along the cell. So for higher sensitivity, many systems will utilize Rayleigh interference optical system in place of alternative Cellerian system. There are a lot of applications of ultracentrifugation. These determine sample purity, you can say, through analysis of sedimentation boundaries, determination of relative molecular mass by sedimentation velocity method and sedimentation equilibration method. They characterize, you can characterize assembly and disassembly mechanism of biomolecular complexes, determine subunit stoichiometries. You can detect and characterize macromolecular conformational changes by differences in sedimentation velocity of the sample. One can measure equilibrium constants and thermodynamic parameters for the self and hetero associating systems. So, there could be a lot of applications of this analytical ultracentrifugation technique. To summarize this lecture, in this lecture we have discussed about different types of centrifuges, right from simple benchtop centrifuge to high capacity refrigerated centrifuge, high speed refrigerated centrifuge, ultracentrifuges, where we have discussed about preparative and analytical ultracentrifuges, and also we have discussed about different optical systems which are employed in analytical ultracentrifuges with their applications. In the next lecture, we will be discussing about different methods of preparative methods which are utilized in centrifugation techniques, particularly preparative ultracentrifugation techniques like density gradient centrifugation or differential centrifugation. Also, we will be discussing in coming lectures the different types of rotors are being used and the care taken for these rotors. So, we will stop here in this lecture. Thank you for your attention. Thank you.